This video is all about why we eat food and how we break down that food. Well, the obvious reason as to why we eat food, besides it tasting good, is because it gives us energy. We would not be able to do our normal life things, take notes, watch this video, go play soccer, unless we ate food. Do you remember carbohydrates give us quick energy, short-term energy? You get a quick burst, you're really hyper, and then there's a crash. Lipids give us long-term energy, so anything with fat in it would give us long-term energy. Well, what is energy? Well, in living organisms, energy is called ATP. ATP stands for adenosine triphosphate, which happens to be the name of my turtle. So next time you come to class, say hi to adenosine triphosphate. ATP is made up of three parts: a base, notice on the right, a sugar, in the middle, and three phosphates, purple, yellow, green, hence 3 phosphates. That's why it's called triphosphate. Tri stands for 3, there are 3 phosphates. You might also notice that this looks a lot like a nucleotide. Remember, nucleotides are small units of a nucleic acid, and a nucleotide had 3 parts. It had a base, a sugar, and a phosphate. Hey, so does ATP. That's not a coincidence. ATP is a nucleotide, except it has 3 phosphates instead of 1. How do we get ATP from our food? Well, when we eat food like polysaccharides or polypeptides or phospholipids, enzymes in our body break it down into smaller parts. For instance, polysaccharides get broken down into monosaccharides, polypeptides get broken down into amino acids, phospholipids get broken down into fatty acids. When these organic molecules get broken down, ATP is created. And remember, ATP is a base, a sugar, and 3 phosphates. This is showing you an example of how our body breaks down food. Let's say we eat a piece of steak or a hamburger, and it's a protein, so it's a big protein, so it's called a polypeptide. Polypeptides get broken down into individual amino acids. ATP is created. How about this example? We eat a piece of pasta, polysaccharide. It gets broken down into the individual monosaccharides, and ATP is created. So how is it that ATP gives us energy? This is a bit complicated, so pay attention. ATP stands for adenosine triphosphate. Remember, the tri means 3, so there's 3 phosphates. When you break off the very last phosphate, it releases energy. So if we break off a phosphate, we now are down to 2 phosphates. We started with 3, now we're down to 2. As a result, the new molecule is called a diphosphate. Di stands for 2. When you break off that last phosphate, energy is released. This is how ATP gives us energy. It starts with 3 phosphates. Our body breaks off the last phosphate, and when it breaks off the last phosphate, energy is released. We will then use this energy to do work. So what kind of work? Maybe we'll think, maybe we'll write, maybe we'll run, maybe we'll make cells, make muscles. Anything that you do, you need energy for it. So we eat food, our body breaks down that food, ATP is created, the last phosphate on ATP is broken off, and energy is released. This happens billions and billions of times every single second in all of your cells. Lots and lots and lots of ATP is constantly being created, and less phosphate is constantly being taken off. Do you remember a few slides ago I said something about how enzymes in our body break down food, but I never actually explained what enzymes were? Well, this is where I explain enzymes. So you first have to understand the foundation before I get to enzymes. Everything in our body is a result of a chemical reaction. A chemical reaction is a process of changing one molecule into another. So if we eat a big piece of, let's say, bread, and that bread therefore is called a polysaccharide, our body breaks it down into a monosaccharide. That's a chemical reaction. What if we eat a piece of fish? The polypeptide gets broken down into amino acids. What if you have a slice of cheese? The phospholipids get broken down to fatty acids. Those are all chemical reactions, DNA being broken down into nucleotides. You could also do the opposite, by the way. You could build monosaccharides into polysaccharides, amino acids into polypeptides. But the point is, whenever you change one molecule into another, that's a chemical reaction. The problem with chemical reactions is it requires a little bit of a push to get started. That little bit of a push is called an activation energy. It's the amount of energy needed to start a chemical reaction. For instance, let's pretend you had a bunch of logs of wood hanging around your house. Would it automatically combust and start a fire? No, it needs a spark. It needs a little bit of a push in order for it to start a fire. Let's say you take a package of Jell-O. You know that powder stuff you pour in water? If you just pour it in water, will it automatically turn into that hard Jell-O? Nope. You gotta stir it, you've gotta heat it, and then it will become Jell-O. The point that I'm trying to make is that the stirring and the heating and the match with the spark is the activation energy. It pushes the reaction to occur. Without the activation energy, the chemical reaction could never occur. This is the point I'm trying to make, that all chemical reactions have a hill to climb. There's a little bit of energy needed to start the chemical reaction. If we're breaking down food to create ATP, we still need a little bit of energy at the beginning of the chemical reaction for it to occur. Okay, so let's bring it back to enzymes now. I showed you this animation before, and I said a polypeptide gets broken down into amino acids and ATP is created. But I never talked about that yellow blob, and that yellow blob is an enzyme. What is the point of this enzyme? An enzyme is a protein that speeds up chemical reactions by lowering the activation energy of the reaction. I know that's a lot. Write it down. They're proteins. They speed up reactions, so they make them happen fast. And the reason they make them happen fast is because it reduces the amount of energy needed to start a chemical reaction. It lowers the activation energy. So it requires less matches, less stirring, less heat, and a smaller hill to climb. You need less of all of that because enzymes are present in the chemical reaction. So for instance, when this happens and you want to put these amino acids together to make a polypeptide, you can do it really fast if you add an enzyme. If you want to break this polypeptide down into amino acids, you could do it really fast if you add an enzyme. Most reactions can occur without an enzyme, but they happen so painfully slow. Like this. Watch. I'm making you watch these purple blobs move across the screen. Look how slow and boring this is. Still watching them move. That would be what would happen if there was not an enzyme. If you were to eat a piece of bread, it would take you so long to digest it. But if you eat a piece of bread, a polysaccharide, and enzymes come in, it could break them down immediately. It breaks them down within a fraction of a second. The point of me showing you all these purple hexagons is to show you that enzymes speed up chemical reactions, and they do so by lowering the activation energy. You need less of a push in order for the chemical reaction to occur. So here's a question, and it's kind of difficult. I want you guys to look at some data. The question is, the enzyme catalase, just the name of an enzyme, breaks down hydrogen peroxide. You know that stuff that comes in those brown bottles at your house? The enzyme catalase breaks down hydrogen peroxide. When was catalase added to the reaction? So I'm going to show you a bunch of data. The data shows the time in minutes. So we have 30 seconds, 1 minute and 30 seconds, 2 minutes, 2 minutes and 30 seconds, and 3 minutes. And here I'm going to move this blue square, and we're going to see how much hydrogen peroxide is breaking down. Looks like not much is breaking down, and then all of a sudden, a lot of hydrogen peroxide is breaking down. Let's read the question again. When was the catalase, which is the enzyme, added to the reaction? Well, when it started working fast, right about here, right about at 1.5 minutes, the enzyme must have been added to the reaction. Before it, really slow. After it, really fast. You could also graph this data. Look at the first minute. Not much is happening. Not many molecules of hydrogen peroxide are being broken down. It's all about 0. But when you add an enzyme, whoa, speeds up really fast, and now the hydrogen peroxide is being broken down really, really fast. The second part of this video is the rest of the enzyme stuff. In order to really understand enzymes, you need to know some vocabulary that helps you understand chemical reactions. Reactants are the molecules that go into the chemical reaction. Reactant, reaction, kind of makes sense. Products are what comes out of the chemical reaction, so whatever is produced, the product. So in this example, we have 3 amino acids. An enzyme comes along and those 3 amino acids are going to be converted into a polypeptide. What goes into the enzyme is called a reactant, and what comes out are the products. Same for the other way around. What goes into the enzyme is called a reactant, and what comes out are the products. So enzymes work on reactants and produce products. But what is special about enzymes is they only work at very specific temperatures. The enzymes in our body only works at our body temperature. Our body temperature is 37 degrees Celsius. You probably remembered it as 98.6 degrees Fahrenheit, which is fine, but science tends to use Celsius, which is 37 degrees Celsius. So human enzymes work best at 37 degrees Celsius. This is called the optimal temperature. The temperature at which the enzymes work best is the optimal. If the temperature is below the optimal, it is too cold for those enzymes to work. They don't work very well, they move too slow, they can't actually function. If the temperature is too high for the enzymes actually fall apart. They do something called denature. They just totally get destroyed and they no longer work. So the point of this graph is to show you that enzymes only work at an optimal temperature. Sometimes this optimal temperature is different, but that temperature is specific for those enzymes. And it happens to be that humans' optimal enzyme temperature is 37 degrees Celsius. Enzymes also only work at a specific pH. Remember, pH is the measurement of acids and bases in a solution. Human enzymes usually work best at a neutral pH because water, which makes up most of our body, and blood, which really makes up the rest of our body, both have a neutral pH. So it makes sense that the enzymes that live in the water and blood work best at a neutral pH of 7. Here's a graph kind of showing it. You'll see that the optimal pH for this enzyme is 6.6 according to the graph. If it's too low, it's too acidic, the enzyme doesn't work. If it's too high, it's too basic and the enzyme does not work. Again, the point is that enzymes have an optimal temperature and an optimal pH. Final thing is the way that we name enzymes is kind of similar to carbohydrates. So notice the enzymes' names are catalase, maltase, sucrase, lipase, peptidase, amylase. All of them end in ASE. I will give you random names of enzymes that you have never heard of before, but you will know it's an enzyme because it ends in the letters ASE. All right, that's all I have. Write down your questions and ask me in class.